introduce Professor Marcos Aguiar from the Institute of Physics at the University of Campinas. He concentrates his scientific effort to study population dynamics in several contexts, and today he will talk about physics and biology.
Okay, well, thank you for the introduction. It's a great pleasure for me to be here. I want to start by congratulating the ICTP-SAIFR and all the people that made this happen. I think maybe the visitors from abroad don't understand how unique this place is. I think this is one of the only places in Brazil where you can really find a very international environment. You can come here anytime. The workshops, the schools are really very well organized, with people from all over the world visiting at any time. So it's really a unique place in Brazil, and I'm very happy to know that this grant from FAPESP has been renewed. So today I'm going to talk about physics and biology here at the ICTP-SAIFR. I've been collaborating with Nathan and other people from about 2 years. And for those of you who really follow the activities of the center, you have certainly realized that there has been substantial effort in trying to organize activities that bring together biologists and physicists. I think the main idea behind this is that this interaction between physics and biology can really bring a better understanding of the living world. The biologists, they have this very detailed knowledge of life, of species, interactions, environments, and physicists, on the other hand, they're always eager to write down equations, to extract fundamental principles, and see what is really fundamental that could explain all this biological mess that we see on the world. Among these activities that we have been organizing here, these summer schools that take place in January, they have been very successful. The students that come here for school, they often return. We ask them to fill evaluation forms, and we get always very nice outputs from them. And so what I thought about for this talk is to talk a little bit about these summer schools. They're very unique also. They are organized in a special way. And after that, I decided to select 3 topics from the school we had this year and talk about them, so that you get a flavor of the type of biology that has been going on and that we have been doing here. So I selected the things that are related to population biology in some way. One has to do with the origin of species, then randomness in biology, and the evolution of cooperation. These were all mini courses that have been taught by people this year. And if I have time, probably I won't. I will talk a little bit about my own work on phylogenetic trees. We will see how it goes. So these are the people involved in this, the organization of these schools. Nathan, Roberto Prenkel, and all these other people. I actually joined the group only 2 years ago. And this is how it goes. These schools, they have a structure that is separated into 2 parts. We have a first week, which is a more hands-on school on mathematical biology, and a second part of the school, which can be another week or more, depending on how we organize it. And the students, they can apply either for the first week, or for the mini courses, or for both. So the first week is very interesting. We usually try to select 48 participants. We are more or less strict about this, from all over the places, but we we usually have lots of applications from Brazil, of course. So we take half of the students from Brazil and half from other countries. Half from biology and half from mathematics and physics. And we divide the students in groups. So we form 8 groups of 6 people. In these groups we have half Brazilians, half foreigners, half biologists, half physicists, and we get teaching assistants to help with the teaching. And the way it works is very simple. There are very few classes. They get one class of 9 minutes in the mornings, which is usually on population dynamics and ecology. And they have all the rest of the time to work on projects. So they have to select, they read papers, they select a paper that, of course, we indicate papers that are appropriate. They pick one they like, and they have 1 week, which is the duration of the school, to work out a model explaining that paper. So they learn about how to write down models, how to program that on the computer, and at the end they have to write a short paper and give a talk about what they achieved. So this is very intensive, and the students are, when they get to the last day, they are really dead. They have worked a lot, but they also learned a lot. It's very intensive and very nice. And then in the second week, we have mini courses. So we invite people to give short courses. I have listed here some of these courses that we did in the past years. So you see the topics, community dynamics, epidemics on networks, spatial ecology, and so on. In 2014, we had the pleasure to have Simon Levin here with us. And this year in particular, we had a very long school. This lasted for 3 weeks. We had 9 mini courses, 3 per week. And this is a list of people who participated. And there were extra seminars that we arranged. And at the end, the students also had to write projects. They worked on projects. And this is the title of the projects they presented at the end of the school. So you see it's very interesting stuff that they did. The origin of HIV pandemics, game theory, speciation, vaccination in game theory. So it's really something that the students get involved. They interact with one another, biologists and physicists, and they really get things done. So, having said this few words about the school, what I thought would be nice for this talk would be to, since most of you are theoretical physicists and probably don't know much about this, I have selected from this list of courses 3 topics, and I will tell you a little bit about them, about the science, what are the questions that they are trying to answer, so that you can get a flavor of the physics and biology interaction here at the ICTP. And so I'm going to talk about speciation, evolution, and the origin of species, as presented by Ulf Dickmann. Then a little bit about microbial population dynamics. This is a beautiful experiment done by Stanislas Liebler. I had to read these papers for preparing this talk. I'm sure if you ask questions, I won't be able to answer, but I think you get the idea what's behind these experiments. And then, what's the other one? It's a game theory, the evolution of cooperation, which is also a very interesting topic. So let's see how this goes. So these 3 things that I selected, they are connected by a common thread, which is population dynamics. And when we talk about population dynamics, the first thing that comes to mind for a person who works in biophysics is the Lotka-Volterra model. This is a very simple model that came up almost 100 years ago to describe the interaction between prey and predators. And it's really very simple. These are the equations. The idea is to describe the growth of these populations as depending on their own characteristics and on the interactions between them. So P is the prey. So the idea is that prey grow exponentially fast, like rats or rabbits. They grow very fast if left alone, but in the presence of predators, you have a decrease in their growth rate because they are eaten by the predators. And the predators, on the other hand, they die out if left alone, right? There is no food, but if they eat, they have a chance of surviving and get a positive growth. So this pair of equations is very simple. It's actually integrable. There is a conserved quantity here that you can calculate so that the phase space is just a bunch of circles. And the populations oscillate as a function of time. Although this is extremely simple, it's an important model because I think it was the first time biology got quantitative. It was a first attempt to write down equations for biology. And you can add lots of stuff there. You can add more species, you can change the interactions, you can add space, properties of the environment, genetics, sex. You can do whatever you want and you get much more interesting results. So, first topic by Ulf Dieckmann, I'm going to use these ideas to describe one possible way that one species can branch into two. Okay? So it's actually might be a little funny to talk about speciation these days. I mean, Darwin had solved this problem 150 years ago, right? It's the name of the book is The Origin of Species, so why do we still care about that? Well, the naive answer for why species arise is mutations and natural selection. But if you think about it, things are much more subtle. For suppose you have a population living on an environment where it's good, it would be nice to have sharp teeth. So those individuals with sharp teeth, they eat more, they reproduce more, so the next generation will have sharper teeth than the previous one. So there is evolution by natural selection, of course mutations guiding that. But that does not explain speciation, that just explains the evolution of a population. So the entire population will have sharper teeth. So where is the branching? So for a population to branch into two, something else must be going on. There must be another element in the evolution to make that happen. So here's an idea. So this is an equation of population dynamics type of equation, but not of a single population, but of a continuum of populations. So I'm modeling, this is my running example here, a population of birds. I have a characteristic, which I call x, which is big size, and n of xt is the density of individuals with that particular big size at time t. So there is a continuum of individuals, different big sizes, and this is this very simple equation. Population grows with an intrinsic rate r, but it's also limited by the amount of food available. Let's call this an island where the birds live. So if the population gets to k of x, which we call the carrying capacity, it stops growing. So you can solve this equation immediately, and what you get is a curve that grows all the way to k of x. And what is k of x? K of x is related to the amount of food available. And we are going to associate that with a certain distribution of seeds that the birds can eat. So let me jump a little bit ahead. So suppose that this is the carrying capacity, so this is the maximum population that the island can sustain for each big size, and it's a Gaussian. So we make a Gaussian model of it. So the idea is that there is a distribution of seeds. There are lots of seeds of this size here. There are less big seeds. There are less smaller seeds. So if you have the proper big size, you have the optimal big size here, which is 0, there will be more food, then you can have more birds of that type in there. And the question is, given this curve, is it true that your population of birds will sort of follow it? There will be lots of birds with average big size and few of the extreme cases. Well, not really. So we have to understand that birds compete. So if you have a certain big size, you eat seeds on a certain range, and someone with a similar beak will compete for food with you. So there is competition for resources. And this is the way it works. Suppose first you have just one big size. So the island is colonized by a certain bird with this big size here. It grows to the carrying capacity, K of x, and then there is a mutation. So a new bird appears with a big size slightly different. So what's the equation for this mutant? It's the same equation, right? It grows with the same intrinsic rate r. However, because there are lots of birds of size x, they are eating most of the food. So this is the meaning of competition. What this bird sees is that there is almost no food left for him, because the x's are the carrying capacity, they are eating everything, so there is very little food left. So here, you don't put just n of y, as we did before, right? Because this is full of birds eating up your seeds. So this is how much you see, the effective population that you see. And what you find is that, so you can analyze this equation. This is all very simple math. And what happens is the following. Oops. So if you're here and your mutant appears with a big size that is farther away from the optimum, it disappears. It doesn't grow at all. Your population disappears. If it's towards the optimal big size, it grows. So if you have mutations going on all the time, randomly, those that are on the wrong side disappear and the population drifts towards the optimal big size. However, when it gets there, something strange might happen. So if competition for resources is very intense, if this width of this competition kernel here is small, the population gets unstable and branches into two. So what's going on here is that it's more advantageous to have extreme big sizes where there is little food but little competition than to be in the middle where there is lots of food but fierce competition for it. And what's interesting here is that if you suddenly, after the branching, if you kill this population here, you just kill all the birds with large beaks, you are back at the beginning. This population will evolve again to the optimal big size and branch. So this means that the fitness of these populations, they are not only the result of the amount of food and big size, it's also a function of the other populations available. So this view, this traditional view that evolution is a population climbing high fitness peak is wrong. Because as the population climbs the peak moves. It moves around. So everything changes. So if you have this big size in this situation, this is very good, you're stable. But if you kill the other part, things change again. So this is illustrated here, so I steal this slide from Dickman. So this is the fitness landscape. This is your evolution of your population. So it evolves towards the optimal big size, but this is the minimum of the fitness landscape, and then it branches. When it branches, you get this to maximum. But once you kill one of these populations, the landscape changes back again, and things restart. So this is a very nice framework to study evolution in this modern perspective, where you have a fitness landscape, you have populations around, and as the populations evolve, trying to climb these peaks, the landscape itself moves. And at the end, you have a completely different situation. So every population reaches its local optimum, but everything here is correlated. Kill one population, everything changes again. So this is very relevant not only to understand the process of evolution, but also for conservation biology. You have to know where you are, you have to know the consequences of wiping out a species on the entire environment. So we had a very nice, so this is the very basic principles of this framework, and we have a very nice series of lectures by Ulf Dickman on this subject with applications to fisheries and stuff like that. So this is one of the topics I want to talk about. I'm going to move on to the second one. Which is a course given by Stanislas Liebler on microbial population dynamics. So the idea here is the following. Biology has a lot of history involved. So we have these external factors like meteorites that fall and kill the dinosaurs, volcanoes and stuff like that. But you also have a lot of internal variations and randomness, like genetic mutations, random interactions that happen. And the thing is that it's, on a more fundamental level, is biology a science or a history book? And Jay Gould asked this question, would intelligent humans evolve again if you could rewind the history of the Earth and reset Earth again and do it again? Would us humans evolve again? And well, of course we can't rewind time and run Earth again, but we can do experiments. And Liebler and his collaborators, they did a very beautiful experiment to test the role of randomness in biology. And this is going to be very brief. I'm going to tell what I understood from the experiment. But essentially what they did is to build a little environment. This is very small, it's a very small glass, with 3 species of microbes that included a green algae. This little thing was sealed and illuminated by light so that the alga could do photosynthesis and generate food. This food would be eaten by the ciliate, and then the bacteria would destroy the residues, and this would be a closed cycle just pumped by the light that you shine on this environment. And they managed to make this and keep it going for 1,000 days. So for nearly 3 years, they could sustain these 3 species of life inside this little tube here, and did several replicates of it. So there were of the order of 20 replicates of this little ecosystem, and they could follow the population of each of these 3 species over time. So what's the idea? If you look, so they describe the experiment in great detail, and basically when you look at each of these replicates, you see different things. There are colonies on the walls, on one vial that you don't see in the other, all kinds of differences that are visual. And if you look just as the population evolves as a function of time, just think you have just one curve here. For instance, this is one of the species. It oscillates, it changes as a function of time. And if you want to study the randomness, the variations of these around some mean, you first of all need to find the mean for then to calculate the oscillations, the variations. So with these 20 replicas, first of all they find that they are very reproducible. So all the 20 samples gave very similar results. This is a time of 100 days. And with this curve they could calculate an average. Given the average, you could calculate the fluctuations around the average, and you study correlations between fluctuations between different species. And what they found in very simple terms is that this these fluctuations are really correlated. I mean, what happens to one species is correlated to what's happened to the other one. So although there are fluctuations, the fluctuations are different for each case, they are indeed correlated. So the interactions that are there between species are playing a very important role. And moreover, you can study these correlations, the power spectrum, and other things. They did a very complete job here. I'm not going to go into the details, but the main conclusion is that despite these wide variations, the three species were always correlated. The correlations emerged and stabilized after 3 weeks and remained so for 1,000 days. Right? And of course, they are coupled by the ecological interactions. So my interpretation of this experiment is that, of course, chance is very important, but you can understand these fluctuations. It's not crazy things that you won't be able to replicate. So interactions do play a role. So biology is a science after all. And here is more detail about how these fluctuations behave. They behave like a sort of random walk with power law behavior. And if you want to read more, I really recommend this paper. It's very nice. It was published a few years ago. Okay, and finally, my last topic, which is the evolution of cooperation. And the question here is very interesting. It has been around for quite some time, and it is why people like us here and other animals cooperate if natural selection is based on competition. So what we want to do is to kill everybody to succeed. So why do we cooperate? And this is not just humans. Right? You have many examples in bees and ants and other societies where people do cooperate. And this has to do with a subject of game theory that started in economics and in biology almost at the same time. And I will say a few words about that. So the main the paradigm of cooperation is the so-called prisoner's dilemma. You have probably seen this before, but I'm going to tell the story again anyway. So the story is as follows. You and your friend committed a crime, and you were caught by the police. So the police want to know what you did, if you're really responsible for the crime, and they are going to question you on separate rooms. So you'll be here, your friend will be there, and the way it works is as follows. If you cooperate with your partner, not with the police, if you cooperate with your partner and don't say anything, say, no, we didn't do anything, then both get 1 year in prison. Because the police doesn't have proof enough, but they are very suspicious, though they are going to help you in prison for 1 year. But if you defect, if you say, no, he did it, and he does the same, he tells the police that you did it, then the police, of course, concludes that you both did it, and both of you get 7 years in prison. And then there is the other case, which is more interesting, that you cooperate. You say, no, we didn't do anything, and your friend says, no, he did it. So what happens is that your friend goes free, and you get 10 years in prison, because you get all the fault. Okay? So this is the so-called payoff matrix. You read it horizontally, and that's the way you read it. If you cooperate and your friend cooperates, you get 1 year in prison. If you cooperate and your friend defects, you get 10 years. Now, if you defect and your friend cooperates, you go free. All the blame goes into him. And if you both defect, you get 7 years in prison. So why is this a dilemma? So why is this called the prisoner's dilemma? The problem is the following. Suppose you are in your cell and you are trying to decide what to do. So you say, well, let's suppose that my friend is going to cooperate. So if my friend cooperates and I cooperate, I get 1 year. Now, but what if he defects? Then if I cooperate, I get 10 years. If I defect, I get only 7. So the logic conclusion is to defect, always. You always defect. And of course your friend does the same. And then you both go to prison for 7 years. Right? So this is the dilemma. I mean, the logical decision is to defect. Then you both get 7 years. But of course the best solution is the illogical one, where you both cooperate and you get just 1 year. So this is the dilemma. And there are several, so this can be generalized. Let me just comment on this. You can have this matrix with other values here. This is usually called the reward, the success payoff, the temptation to defect, and the punishment. And if you do a population dynamics analysis of this, this is how it goes. So suppose you have a population and you want to know the fraction of cooperators and the fraction of defectors in this population. So these are fractions, they add to 1. Oops. So if this payoff, this reward, all these numbers that you put here, you translate into fitness. So you can write an equation like this. So the growth rate of the cooperators, so it's the number of cooperators times the fitness, it's the growth rate of cooperators, and this is how you calculate the fitness. If you interact with another cooperator, which happens randomly with a proportion XC, it's a fraction of cooperators, that's the chance that you run into a cooperator, you get R. But if you interact with the defector, you get S. And same thing for the defectors. Okay? And you can show that in this type of prisoner's dilemma thing, the fitness of the defector is always greater than the fitness of the cooperator. The defectors dominate the population and they win. There is no way you can start a cooperation here. So there are many ways of this puzzle here. So how can you change things in order to give cooperation a chance? So I'm going to show you one of the ways. And it's as follows. So this is kind of interesting. It's again very simple, but it leads to an equation that became famous. So one way out of the dilemma is to get the cooperators a chance to interact more with cooperators than with defectors. So if you can choose who you interact with, then cooperators should interact more with the cooperators where they get more instead of interacting with the defectors. So suppose you have this payoff matrix here. So you're going to cooperate. If you cooperate, you get a bonus for cooperating, but you pay a cost. Cooperating also always involves a cost. If you cooperate in the other defects, you pay the cost and get no benefit. All the benefit goes to the defector. And if both defects, you don't get anything. So this is just a version of that other matrix. And I'm going to define the probability that an individual chooses to interact with another individual. Okay, so it's not going to be random anymore. I'm going to define this number R, which is the probability that you choose to interact with an individual that is identical to you. Okay? And 1 minus R is the probability that you pick an individual at random to interact with. So X is a fraction of cooperators, 1 minus X is a fraction of defectors. So this is how it works. So the probability that a cooperator interacts with another cooperator is R, because it chooses to do so. And the rest of this probability is 1 minus R times the fraction of cooperators. This is the thing that happens by chance. Defector with defector is again R. It chooses to interact with its equal. And then 1 minus R times the probability that interacts by chance, and the number of defectors is 1 minus X. The others you can calculate by complementarity here. C with D is 1 minus C with C, and so on. And then you calculate the fitness functions. So what's the fitness function for a cooperator? So if a cooperator interacts with a cooperator, you get B minus C. If you interact with a defector, you get minus C. So you plug these numbers in, and this is what you get. Same thing for a defector. Defector with a cooperator gets B. Defector with a defector don't get anything. So now, if you impose that the fitness of the cooperator is larger than the fitness of the defector, you get this very simple relation. Just plug in all these numbers and you get BR larger than C. So this is called the Hamilton's rule. And it says that the benefit for cooperating times this parameter R has to be larger than the cost. And this R has been often interpreted as kinship. So if you cooperate with your brother carries half of your genes, right? So the chance that you're going to interact with him is 1/2, because you want to save half of your genes. If you interact with a cousin, the cousin carries 1/8 of your genes. So this R is smaller. You see that if R is 0, there is no way that cooperation will evolve. 0 cannot be larger than C. C is positive. And this led Hamilton to a very famous phrase that he would jump in the river to save 2 brothers or 8 cousins, not less. Right? It's not worth it, right? So, okay. So this is how this field of cooperation goes. So this is one way out of the puzzle. I mean, to interact not randomly. There are many other ways to do it. You can interact many times. If you run into a person multiple times, then if he defects on you, he will know that. Next time he will act differently. So there are many ways to do that, and this is just one just to illustrate how the field goes. Okay, I'm about to finish. I was going to say a few things about my own work, but I think I won't have time. So this is the topics I told you a little bit about. There are others which are really fascinating. There are neural networks, how the brain works, how consciousness arises. There is emergency. Bill Bialek also gave a very nice series of lectures on emergency, on biological systems, pattern formation, biomechanics, cancer, hematopoiesis. So there is a whole new areas of biology where physicists can put their hands on and give contributions. So I think these schools that we have been organizing here has been really a wonderful opportunity to meet with all these people, talk to them, exchange ideas. The students have been cooperating a lot. They have been working on projects that have been suggested by these people. This works in terms of arranging for postdocs and so on. So it's been for me a great honor to participate in this organization and also to be here today giving this talk. So I want to finish by once again congratulating the Institute, and I wish you the best for the next 5 years. So thank you.
So we have time for some questions.
Also for David Cross, if you...
How does this research is received by the biology community? Do you publish in physics journals or biology journals?
Well, I will speak for myself. I've been trying to publish always in biology journals. If you, depends a little bit on the topic. Sometimes you get something which is very formal. For instance, right now I'm working a little bit with the topology of phylogenetic trees, and there are some very interesting mathematical properties that you can analyze. So I'm not sure I can send that work to a biology journal, but usually we publish in Evolution, the Journal of Theoretical Biology, journals that are into biology. Otherwise, you don't get read. I mean, if you publish this in the Physical Review, your work gets lost completely. And there is also an art in writing for a biology journal. It's totally different. It's totally different. My first papers were just refused by the editor. So, there is no discussion here. Discussion in biology is 3 pages. I mean, results is 2 or 3 plots. And then there is a whole session of discussion that we are not used to do that. So we have to get into this philosophy, which is interesting. I mean, it's not wrong. It's the proper way to do it for biology.
Yes, hi. Can you briefly, if it's possible, tell us about your work, ongoing work?
Okay, I can show one figure and just to illustrate. Okay, here's the thing. So first of all, how do you know that the species that you see today are really related? So what you do usually is that you study the morphology and more recently the DNA of these species, and you make this assumption, which is very reasonable, that the similar they are, the closer in time they are related. So by doing that, you can construct these phylogenetic trees, where here you have species, this is time going backwards, and these are the times where the species have branched and speciated. And the question is, how much information is there in a phylogenetic tree? You have the times of the branches, you have the order of the branches, who branched with who, but can you say something about the process that generated this speciation event here? Was it by geographic isolation, sexual selection, competition for resources? Can you say something? And the idea is that if you look at more simple trees, this I just drew myself, but if you see a tree like this, which is very asymmetric, and a tree like this, which is much more symmetric, I mean, the process that generates this tree cannot be the same. There must be something about the process that is connected to the topology of the tree. And so we've been studying that, and to do that, first of all, we have to ask, given, for instance, here I have 8 species, how many topologies are there for each species? You can calculate that is well known, but it's a kind of complicated expression. And the number of trees grows exponentially fast, and you can actually quantify the topology. It's a simple quantification, but you can do it here. It is. It's called the Sacking Index, and this is the way to do it. So this is the 2 possible trees for 4 species. What you do is that you count the number of nodes all the way to the root. So for this species you have 1, 2, 3, for this 1, 2, 3, for this you have just 1, 2, for this you have just 1, and you add them up. This is an index. And you can show that the asymmetric trees have higher index than the symmetric ones. So it's a way to know where you are in this spectrum of trees. And we have been doing, last figure, we have been doing simulations with a particular model where we change parameters. Here we are changing the size of the genome that we insert in these individuals in the simulations. And we can show that if the genome size is small, we get a very asymmetric tree. And in this case, The spatial structure of the population plays a very fundamental role. The species they are born in separate regions in space. We have a very high correlation between genetic space and spatial space. But as you increase the genome size, this correlation decreases, and if the genome is infinitely large, you have no correlation at all. Speciation happens without space. And what we have been seeing is that the Sacking index changes considerably. So these are simulations where space is very important. These are simulations with infinitely large genomes, and this is something in between. So there is some signature of the process of speciation in the tree. So we are studying that right now. We are trying to include other types of processes to see how this changes, and then eventually go to data and see if we can say something. But that's the idea.
Okay, so we thank both the speakers. So now it's coffee break time, and we will start at 5 p.m. Okay.